Hi, my name is Sophia Marshall-Pelayo, and my research is biopolymer-stabilized lutein emulsions and their electrospray drying. I'm doing this research under Dr. Kong, who is in the Department of Human Nutrition, Hospitality, and Sport Management here at the University of Alabama. To begin, what is lutein? Lutein is an antioxidant and carotenoid, or pigment, that's found in leafy greens and orange or yellow vegetables. Its health benefits include promoting eye health, enhancing cognitive ability, and reducing the effects of LDL cholesterol or bad cholesterol in the body. Lutein is unable to be synthesized by humans, so the body relies on dietary intake. Why is lutein important? It is concentrated in the macula of the eye, where it acts as a blue light filter and guards the macula against light-induced oxidative damage. Lutein is the primary component of the macular pigment, and as an antioxidant, it protects cells from damage caused by unstable molecules called free radicals. So lutein lowers the activity of these free radicals in the macula, so they don't cause as much damage. Newborns have a higher permeability in their retinas, so they're more vulnerable to this light-induced oxidative damage and have the most abundant carotenoid in the infant brain is lutein. This brings me to our focus on lutein in infants. So around 14 weeks of gestation, lutein accumulates in the vitreous humor of the eye, and around 22 weeks of gestation, this accumulation peaks, suggesting that lutein plays a large role in the development of the retina. Lutein also has an inhibitory effect on the progression of retinopathy of prematurity, or ROP, which is the leading cause of blindness among preterm infants. Preterm infants also have lower serum and brain lutein concentrations, especially formula-fed preterm infants compared to those that are breastfed. There was a study done where preterm infants were fed for 4 months with formula fortified with lutein, and they showed greater rod photoreceptor sensitivity and faster neural response. But once you add an emulsifier to the equation, they create a stable mix or an oil and water emulsion, which is small lipid droplets dispersed in an aqueous solution. Emulsion-based delivery systems are believed to slow down the degradation of carotenoids and enhance their bioaccessibility. Polysaccharides are good emulsifiers for emulsions for food. Which brings me to OS starch, or the emulsifier we are using, capsule TA or CTA. OS starch is food-grade starch that has been modified with octenyl succinic anhydride to have both hydrophilic and hydrophobic properties. Capsule TA, or CTA, the specific emulsifier I'm using, is modified starch derived from tapioca. CTA is often used in the creation of dry food products that are going to be rehydrated with liquids, things like dry cake mix. This is how we get the dry powder from the emulsions, electrospraying. So in electrospraying, the end goal is to deposit a dry powder. High voltage is applied to the liquid, which is dropped from the syringe, and breaks it into a fine spray of highly charged liquid droplets, which end up becoming dry powder. Our goal is to get dry powders from the emulsion that can be rehydrated with liquids to make things like formula. Dry powder is also more cost-effective for transportation and distribution compared to liquids, which determined that using 20, 30, or 40% ethanol when dissolving the CTA to make the water phase actually reduced the interfacial tension and increased emulsion stability, so I'm now using 40% ethanol when creating the water phase. The next is homogenization, which I'm keeping constant at 20,000 RPMs for 2 minutes. The voltage is also being kept constant during the electrospraying at 25 kilovolts, and the flow rate during electrospraying is also kept constant at 0.05 milliliters per minute. Our goal is for the emulsions to be more fluid rather than gel-like, and I'm currently using safflower oil instead of the lutein ester. On the left is a picture of the emulsion after it's been homogenized, and on the right is our electrospray setup. So on the bottom is the foil that's ultimately going to receive the dry powder emulsion, and at the top the red arrow is pointing at the needle, which is really hard to see, which drops the emulsion from the syringe at a flow rate of 0.05 milliliters per minute. The left is a photo of the dry powder emulsion once it's on the foil. This was taken right after we finished the electrospraying, and as you can see, the white section compared to the more foil shiny section, the white is going to be the dry powder that we receive from electrospraying the emulsion. On the right is a photo of the dry powder after it's sat on the foil for about a week. And as you can see, it's a lot harder to see than right after we did the electrospraying. The red arrow is pointing at the sort of line where you can see the division between the foil and the powder. We are currently struggling with getting the dry powder off of the foil for further analysis, so we're working on ideas to make that process easier. For the future, we want to use lutein ester to make the emulsions, of course, and then, as I said earlier, determine how to extract the powder from the foil for further analysis. And for this, we're thinking of using parchment paper instead of the foil so that we can get it off easier. And eventually, we want to electrospin nanofibers for lutein delivery. And since lutein and OS starch cannot be electrospun on their own, we're going to need to add a fiber-forming polymer to aid in this process. Here are my references. Thank you. Any questions?
Can you go to slide 8, I think it was?
Yes.
Yeah, so your focus was on initially like for young children and infants. I'm assuming that there's also benefits for supplementation of lutein with adults. So what I'd like you to help me understand is these concentrations here, the emulsifier, the percentages and so on, that you did for the younger children, the infants. Would that be the same concentration for adults? And how did you arrive at 10 and 30%? So where did those numbers come from? And would it need to be changed if we're doing it supplementation for adults?
As for the supplementation for adults, I'm not sure, but that might be answered by the fact that for the CTA, the emulsifier concentration, 20% is the optimal concentration for creating the most stable emulsions based on previous research and previous research in our lab as well. And that's going to be the same for the oil phase volume fraction. So that basically means we're doing 10% oil phase and 90% water phase, and 30% oil phase and 70% water phase. And those are also, they have been found to be the optimal concentrations to create the most stable emulsions possible.
Why are you making the samples with safflower oil instead of the lutein ester first?
So lutein ester is very, very light sensitive. So I am first learning how to make the emulsions and do the electrospraying to the best of my ability with the safflower oil so that I'm not wasting lutein ester on basically practice rounds and getting to learn the process. So eventually, once I'm very comfortable with creating the stable emulsions and electrospraying them well, that's when I'm going to be able to use the lutein ester.
Are there other starches that can be used to make emulsions?
Yes, so there are other starches that can be used to make emulsions. I'm currently trying with CTA because it has been shown to prevent lutein degradation better than some other starches, and it's the best, one of the best starches for the electrospraying process that we're doing with the emulsions.